is my pleasure to introduce Dr. John No, who is an assistant professor in the BME department at Boston University. He did his PhD at Caltech with Dave Terrell and then did a postdoc with Roger Chen. And he has been at BU for 6 years, 5 years?
X number of years. Yeah.
And we're very excited to hear about his awesome work.
Thanks for the introduction and the invitation, Ariel. I think senior graduate students will understand that X numbering designation at some point along the graduate career. Great to be here today. I taught my last class of the semester this morning at BU, and so it's a very special day for me today. The end is near, summer is just around the corner, and I'm glad to be able to tell you today about the research that we're doing in the lab. So once classes are over, we're back into the lab and carry out more work on how to engineer cells to respond to mechanical forces. Okay, so the goal of this work is to train cells or to be able to program cells to be able to sense specific mechanical fields and then carry out a specified biological response in turn. And a few examples of why we want to do this. The first one comes from everyday life. For instance, when we're at the grocery store picking out produce, which I don't do as much anymore now because of all this delivery stuff, but one might test individual avocados in order to identify the one of the perfect stiffness or ripeness and ready to consume. And you may do this at Whole Foods or Trader Joe's, wherever graduate students are shopping these days. And this is an indication as to what type of avocado you might be biting into. Okay. Well, cells carry out the same type of sensing processes all the time, but instead of feeling the stiffness of an avocado, cells will explore the physical properties of their surroundings, including the Young's modulus of the extracellular matrix, so on and so forth. An example that I like to bring up is one that was published when I was in graduate school that caused quite a bit of excitement in the lab. And that is a finding reported by Adam Engler when he was a graduate student with Dennis Disher. And the finding was that mesenchymal stem cells can differentiate down different lineages depending on the stiffness of the extracellular matrix surrounding. So matrices can be isolated from brain, muscle, or bone type tissues in the body. And then if you decellularize those matrices and then grow stem cells on them, there's some information within those matrices that are able to guide these differentiation events. But beyond just stem cell differentiation, we know that stiffness is a biological parameter that can guide cellular events, but also can be an indicator of, for instance, disease. Somebody might go to the doctor because they felt a lump, and that may cause a suspicion that there's a solid tumor forming. Of course, the doctor will do a biopsy, a biochemical analysis to confirm that that lump is malignant or not. Okay? So we know that we carry out this type of mechanical decision-making all the time, whether it's at the grocery store, whether it's stem cells in a dish, or whether it's a doctor and a patient. And so the question that we have in the lab is to understand how this relationship is carried out by cells. That is, how cells sense mechanical forces and how those mechanical forces can then trigger some sort of biochemical or gene expression change. In particular, we're interested in engineering this relationship. So we can think of this input-output relationship being mediated by some sort of transducer, something that can sense a mechanical force and then change that signal into something like a gene expression output. Okay. And so in life, the transducer is, in many cases, signaling proteins. And similar to how chemical signaling events can be mediated by proteins that bind ligands of various different receptors, et cetera, proteins can communicate mechanically stimulated events by changing their three-dimensional conformations. And here's an example of a generic protein. Say, for instance, I wanted to apply force on it across the protein. This particular polymer, I could pull it at either end, and the introduction of that energy across the polymer may deform it from its native state. And in this case, the deformed state of the protein may cause some biological outcome. Okay? So there's several famous examples of how force-mediated protein unfolding can trigger biological responses. You may have heard of integrin activation, but also, in addition to that, very relevant to this year's Nobel Prize are mechanosensitive ion channels that alter their conformation in response to mechanical tension across the plasma membrane. And as a result of this pulling in the axial direction, can allow ions to flow through across the membrane into the cytoplasm. Okay, so in a synthetic biology pursuit, we began to ask how we might take advantage of this mechanically mediated protein unfolding event in order to program cells to sense and respond to mechanical forces. And so we did consider adapting things like integrins or piezo. However, we wanted a more direct relationship between mechanical force and a specifiable output. So for instance, if I return back to this slide here, although one could imagine mutating piezo in order to have different force sensitivities and then to allow ions to flow in response to different magnitudes of physical force, how we could translate calcium fluxing directly into a specified output was unclear, or at least at the time seemed challenging. So in order to program this relationship between mechanical force and biochemical action, we turned to a different protein system and identified a transducer in the form of what's now my favorite protein called Notch. And so I'll tell you a little bit about Notch and how we're using it now to program this relationship. But before I get into that, I just want to point out that most of the work that I'll tell you about today was carried out by this individual here, Chris Sloas, who is now a scientist at Charisma Therapeutics down in Philadelphia, Pennsylvania. And this is a photo from our first time we went to Notch Gordon conference in the summer of 2016. Okay, so Notch is an endogenous signaling protein that is conserved throughout the metazoan family of the tree of life. And it gets its name from its discoverer, who identified a mutant in Drosophila that exhibited a notch in the wing. And this notch here is formed due to mutation of a transcellular signaling protein, which we now know as Notch. And Notch actually, it turns out, is a mechanoreceptor. And it has 3 regions, and we can divide them up as follows. Notch lives at the plasma membrane at the cell surface, and it has a ligand binding arm. And this ligand binding region serves the function of sensing ligands that are expressed on the surface of adjacent cells. And the intracellular side, Notch has a transcriptional activation domain that sits at the plasma membrane in the quiescent form of the receptor. So in the off state, this protein ties up a transcriptional activator at the plasma membrane where it's outside of the nucleus. And in between these 2 regions, there's a mechanosensitive domain that we call the negative regulatory region, or I'll abbreviate it as NER, or the NRR. And so Notch signaling is activated when a ligand binds the receptor via the ligand binding domain, but it turns out that biochemical binding event is not enough to activate the receptor. So if I were to take soluble Notch receptor and apply it to Notch expressing cells, I could confirm that the ligand was bound to the receptor, but signaling would not be expected to occur. The natural stimulus for Notch signaling requires both the binding of the receptor to its ligand and then the application of mechanical force. And this typically happens by the endocytosis of the bound ligand into the neighboring cell. And that endocytosis event delivers mechanical energy to the system, and it causes unfolding in this regulatory region, which then exposes previously hidden protease sites, which are cleaved by cell surface proteases in a 2-step fashion that ultimately results in the liberation of this transcriptional regulator domain, which can then go into the nucleus and turn on its favorite gene. That last cleavage event is actually mediated by a transmembrane protease called gamma secretase. Gamma secretase is a polypeptide component enzyme that lives in the plasma membrane. And I like showing this image because it gives insight into the mechanism of how this signaling protein becomes processed in a ligand-dependent way. So in the quiescent form of the receptor, when signaling is off, there's a bulky group here right at the juxtamembrane position that prevents entry of the protein into gamma secretase. But when we reveal that protease site, cell surface metalloprotease cleaves here, and then we get a transmembrane stub that can now diffuse into this barrel, this enzymatic chamber of gamma secretase here. And so if we look at this green protein is a component of gamma secretase. It's called nicastrin, and it serves a gating function of limiting what can enter into the catalytic chamber. And so if we have a bulky domain right at the cell surface of the protein, it cannot diffuse into the chamber. But once we mediate that first cleavage event, we get this stub that can now readily enter and be cleaved at an intramembrane position to liberate what we call the ICD, the intracellular domain. So while we were carrying out this work, a very exciting pair of papers came out from UCSF, from Wendell Lim's lab, in which they showed that the Notch protein can be reprogrammed or reconfigured in a way that's highly customizable. So I told you about Notch, how it has this general organization. It turns out that you can remove, though, the extracellular domain and the native intracellular domain of Notch and replace it with whatever you want. And in this way, one can reprogram the biochemical input of the receptor as well as the transcriptional output of the signaling pathway by fusing, for instance, an antibody to where the ligand binding domain would originally be, and then a customized transcription factor as utilizing that as the intracellular domain. Okay, so at this stage, we thought Notch has this great programmability. We can define what signals can be put into the signaling pathway and what outcomes can be achieved by swapping the ECD and the ICD. But when we really consider the system, we recognize that Notch actually has two inputs. One is mediated by the ligand binding domain, which determines the biochemical information that the cell must sense and receive. But there's also that mechanical requirement. So as I mentioned, soluble ligand does not activate Notch. That's true for regular Notch and synthetic Notch. So in recognizing that two inputs are required, we asked now if we could fine tune the second input. So the first input, the biochemical input, is easily tunable by swapping out that extracellular domain. But how can now we program the mechanical sensitivity of this region called the NRR? Okay. So this, it turns out, ended up being the transducer that we selected for our engineering scaffold. It's something that I mentioned can be customized and is highly modular. So for instance, I can define the input and output along this pathway. And now we want to fine tune the mechanical sensitivity of this region here to couple specific magnitudes of force with defined outcomes. Okay. So previous studies, single molecule and force spectroscopy based methods had been applied to study the mechanosensitivity of the native human Notch 1 NRR. And a variety of studies all converged on about 5 piconewtons of force as setting the mechanical threshold for what must be applied to the system in order to induce that activation event. And when we began the work, we recognized, 5 piconewtons of force, that's not very much. And we have the option to either tune the force sensitivity up or tune it down. Well, we knew from existing work in the cancer literature that a Notch actually is frequently mutated in this region, and that the destabilization of this domain tends to lead to spontaneous malignant signaling that's independent of force and ligand. So we thought, with that in mind, that fine-tuning the force sensitivity up might be the way to go. So our logic at this point was, if Notch has this 5 piconewton mechanosensitivity, that sensitivity or that force activation threshold may be defined by the interface or the molecular interactions that exist between these various green and blue domains. And that the easiest way to increase the sensitivity of the receptor would be to simply introduce more intramolecular interactions. And it turns out, because this domain or this region of Notch is mutated so frequently in T-cell lymphoma, that researchers at Genentech and other venues have developed antibodies that are able to recognize this domain of Notch and clamp it into its auto-inhibited conformation. So our first venture into tuning the sensitivity of this receptor was to take that antibody and fuse the sequence right into the domain at the level of a SynNotch receptor. And our hypothesis was that this fusion event would introduce additional molecular interactions and therefore raise the threshold by requiring more energy to break those hydrophobic interactions, hydrogen bonds, what have you. So from here on out, I'll compare data from cells that express either a regular SynNotch receptor containing the native, what we call NRR, or cells containing various versions of our now engineered mechanosensitive domain, which I'm referring to as a SNRR, so a stabilized NRR or a strengthened NRR. Also, before I go on, I want to point out that a key aspect that facilitated this work was the availability of high-resolution crystal structures that allowed us to see exactly where all the individual residues were with respect to one another in the antibody-bound NRR complex, and these were available in the PDB. So we designed our receptors using DNA synthesis, and we expressed them in cells. And the first thing that we wanted to do was to confirm that the engineered receptors exhibited the basic properties that would be required for signaling. And if a receptor is going to sense an extracellular signal, of course, it has to get to the cell surface. We also wanted to confirm that our design, our polymer folded as intended with this integrated antibody fragment binding in an intramolecular fashion as depicted here. And then we wanted to confirm that the receptor could be activated, period, because after all, we fused an inhibitory antibody to this chimeric protein, this engineered protein. And we wanted to confirm these three aspects before proceeding with fine-tuning that interaction. Okay, so in order to confirm that the receptor got to the surface, we did a simple immunostaining. These constructs, in addition to all these parts, express an extracellular MYC epitope tag, and we simply took fluorescent anti-MYC antibody and then exposed the antibody to live cells that had intact plasma membrane. The antibody should not diffuse past the plasma membrane, we should only capture what's at the cell surface. And turns out cells that express either the NRR containing SynNotch or the SNRR containing SynNotch lit up in green when probed with the anti-MYC Alexa 448. To confirm that the protein folded it upon itself on an intramolecular manner like so, we used a second antibody and stained the very same cells here. And in this case, we used an antibody that bound the NRR at the same site with the rationale that if this spot were blocked by the integrated antibody, cells expressing the SNRR receptor should be non-reactive to this exogenous antibody. And that antibody carried a distinct fluorophore that we could detect independently. And when we compared the two cells, here's what we saw. So cells that express the NRR containing antibody lit up with both green and magenta, indicating that the receptor was at the surface and that the NRR was accessible. However, cells that express our SNRR receptor exhibited surface reactivity, but lost reactivity due to against this NRR antibody, suggesting that the protein was exhibiting the intended conformation when expressed on the surface of cells. Now we fused this antibody to it, and then we just wanted to make sure before we went ahead and did single molecule force measurements that the SynNotch receptor could be activated, period. And so what we did here was to apply the conventional SynNotch or Notch activation assay. And as I mentioned, soluble ligand does not supply the necessary tension to activate the receptor. So what's normally done is that a ligand for natural Notch or synthetic Notch is absorbed to the surface of a plate. And that absorption event allows the ligand to apply tension to the receptor. And that pulling energy can then induce those conformational changes that leads to the release of the intracellular domain. So in this case, we're using anti-fluorescein as a ligand binding domain here. And so we simply grew our receptor expressing cells on surfaces coated with fluorescein conjugated BSA. And so if signaling were to ensue, we would activate the release of our transcription factor, and we would expect now to be able to detect the expression of some reporter gene. In this case, we used a luciferase-based readout. Okay? So the transcription factor that's cleaved from the receptor goes into the nucleus and turns on luciferase. And so when we grew cells either on control surfaces or cells coated with fluorescein, here's what we saw. In the absence of ligand, so just control surfaces, we saw minimal amounts of luciferase activity. However, when cells were expressing either NR or SNRR-based receptors, when those cells were grown on fluorescein-coated dishes, we observed an increase in the expression of the luciferase enzyme. We're also able to knock down the levels of luciferase activity by inhibiting the proteases that are involved in cleaving the receptor at those two positions that I mentioned before. So together, these data confirm that the receptor can be activated, even if it's activated a little bit less. It shows that the receptor is activated in a ligand-dependent manner and that the activation proceeds in a manner similar to Notch. It requires gamma secretase activity and atom 10 activity, so on. Okay, so we confirmed these three aspects, that the receptor was at the surface, protein was bound in the right conformation, at least we hoped, and that we could activate the receptor to some extent. So now the big question was, great, we can activate the receptors, but how much force now is required to stimulate this receptor relative to the native natural NRR domain? In order to measure the amount of tension that's required to induce signaling, we borrowed a technique from TJ Ha's lab called the tension gauge tether method. And in this approach, what one does is one takes a ligand and one attaches it to the surface of a cell culture dish, but using a bond that can be broken. So this ligand here, this green square, is attached to cell culture dish via a mechanically labile bond. The receptor now, the cells are placed onto that surface. The receptor can engage with the ligand. And then now we have a tug of war. We have a tug of war between two mechanically labile structures. And depending on which structure can endure more force, we'll get two signaling outcomes. So if our tether that attaches the ligand to the surface is a weak one and cannot supply enough tension to break the protein domain, that tether will break and will release our ligand into solution, and then we get no signaling from this particular cell. Now, if we use a mechanically strong tether, that tether should be able to endure, and if it can endure more force than is required to break the protein domain on the receptor, we should unravel that domain there, and then the proteolytic cleavage sites should follow, and then we would get activation of some reporter gene. And in the images I'm about to show you, we use M-cherry as fused to histone instead of luciferase. And the tethers that are actually used to attach the ligand to the plate are actually simple double-stranded DNAs that we order from IDT. They're all of the same sequence, more or less, and they all are the same number of base pairs. And by comparing different tethers of the same, more or less, sequence composition, but of different threshold mechanical sensitivities, we were able to suss out exactly the tension that was required to activate these receptors. And here's what we did. So borrowing the technique from TJ Ha's lab, we took DNA strands that associate with one another like so. We added a biotin handle on one strand in order to attach the tether to the plate. And on the hybridizing strand, we attached our fluorescein ligand. And this actually, fluorescein turned out to be a convenient choice because we could order the oligos directly from IDT, modified with fluorescein, and they show up in a couple days. But by varying the position of the fluorescein relative to the biotin, we could define the amount of tension that was applied to the receptor. And that variation was as follows. First of all, I should mention that all of these tethers have been individually calibrated using AFM. And in this orientation where the biotin is on the same side relative to the fluorescein ligand, it takes about 12 piconewtons of force to break these two strands apart. However, when you have the same sequence of DNA, the biotin and ligand are now, the ligand and biotin are oriented on opposite ends across this double-stranded DNA. Folks have shown that more force is required to break this orientation apart. And you can imagine this as being the difference between an unzipping mode to break these two strands apart versus a shearing mode in order to break all of these bonds interactions simultaneously. And by varying the position of the biotin along the tethering strand to these various relative positions, we can now span the range of 12 to 54 piconewtons. And then now we're hoping that this set of TGTs would inform us as to the mechanosensitivity of our SNRR receptor. So as I mentioned, the NRR, the natural NRR from Notch 1 takes 5 piconewtons of force to break. And folks actually use this to first characterize that threshold. And so for a receptor that takes 5 piconewtons, if you were to challenge those cells expressing that receptor against all these TGTs, since 12, 23, et cetera, are more than 5, you expect that ligands tethered to the surface via all of these designs would be able to stimulate signaling. Since we hypothesized that our SNRR domain was stronger than the NRR, we'd hope that somewhere within this dynamic range would give us a pattern like so, where the weaker TGTs would not be able to supply enough tension to activate the SNRR domains, but something where like in the 54, 50, 43 piconewton range, we'd be able to break that antibody away from its antigen and induce signaling. And we had good reason to believe this to be the case because single molecule studies of antibody-antigen interactions have suggested for many different antibodies that antibodies engage with their antigens with a mechanical sensitivity of tens of piconewtons. So I'll show you some data in terms of what we saw when we grew cells that express these various receptors on these different TGTs. I'll represent the data in 2 ways, and I'll compare the NRR-containing receptor with the SNRR-containing receptor. We first carried out control experiments where the cells were grown in the absence of ligand, and we quantified using flow cytometry the expression of the reporter gene, and we also imaged the cells directly to look for that M-cherry fluorescence. And in the absence of ligand, we don't get much signaling from the NRR-containing receptor or the SNRR-containing receptor. So great, we need ligand in order to activate the signaling pathway. Now, since 12 is more than 5, we anticipated that the 12 piconewton TGTs should stimulate the activation of the pathway and get M-cherry fluorescence. And that is indeed what we saw with the NRR-containing construct. The SNRR-containing construct, though, didn't give us much activation when challenged with 12 piconewtons. So we were happy, but also nervous, hoping that, that the reason for this was that we were at a sub-threshold level in terms of mechanical force. Moving up along the line, 54 piconewtons, of course, gave us plenty of activation for the gene expression reporter here. And we're happy to see that at 54 piconewtons of tension, we were able to get some amount of signaling via this synthetic receptor. Now, the upper sort of limit for our tension application here, instead of using a double-stranded DNA to tether the fluorescein to the cell surface, we simply use biotin fluorescein just to attach it to a streptavidin coated dish coated with streptavidin at the bottom. And streptavidin biotin bond can endure about 100 piconewtons of force or more, depending on who you ask. In any case, it's stronger than all of the double-stranded DNAs. And so under these conditions, we get more activation from both. We get about the same amount of activation from the NRR-containing receptor, but this increased amount of force gives us a bump in the amount of signal that we get for the SNRR expressing cells. Okay, so at this point, we wanted to tune the receptor. We confirmed that it could be expressed. We confirmed that more force was required to activate the receptor, something in the regime of 50 piconewtons. So we went from 5 to 50 piconewtons. And so that's a big spread, a 10-fold difference in mechanosensitivity. Our next question was to ask whether we could populate the space in between. Okay. So here's just a schematic. You could imagine the rupture strength of the natural NRR having an average value of 5 piconewtons here, and then our antibody fused version stabilized the receptor mechanically in order to elevate its rupture threshold by about 10-fold. We asked now if we could fine tune and populate the space in between to get a range of receptors that are all tuned with sensitivity within the physiologically relevant regime. I should mention that endosomes apply about 10 piconewtons of force, and the activation of integrins require about 43 to 54 piconewtons of force. So we're working within the regime that's relevant to cell biology. Okay. So taking a lesson from Notch cancer biology, in which the native NRR is frequently mutated in T-cell lymphoma, we asked whether we could introduce similar mutations in order to destabilize not the NRR in this case, but the SNRR. So we imagined if the mechano-active site or the mechanically sensitive region that defines Notch's activation threshold as being this interface shaded in yellow here, we asked whether now for the sNRR domain, whether the interface between the antibody and the NRR would represent this synthetic mechanosensitive region. And we examined the interface, the nature of those interfaces using the crystal structures available in the PDB, and figured out mutations where we could fine tune the strength of this domain. I won't take you through all the mutations, but I'll take you through a set that makes for good logic or communication in terms of how these things actually work. So they taught us a lot about how this particular domain can sense and transmit force-based information. Okay. So the structure I'm showing you here over on the right is a close-up of the NRR domain, a region of the NRR. And shown in purple here is actually the antibody fragment that we've directly fused to the receptor. And there's an interaction between this tyrosine side chain that forms two hydrogen bonds with the backbone units of the NRR polypeptide chain. And we picked this region to mutate because there were two obvious mutations that we could make that we anticipated would destabilize the interaction between the SCFE, the antibody fragment, and the NRR. Okay? So I'll walk you through the data. First, I'll show you what we saw just with sNRR, now representing it in the form of flow and using, instead of H2B M-cherry, just a regular M-cherry soluble red fluorescent protein as a reporter. So in the absence of any ligand, we get some baseline expression or a level of fluorescence here. And that level of fluorescence, the signaling is the same between sNRR cells or cells that express this mutant sNRR here, where we've mutated the tyrosine residue to phenylalanine. So for those of you who need to brush up on your biochemistry, the tyrosine to phenylalanine mutation involves simply the deletion of this paraphenyl position right here. So we eliminate this OH, and therefore we get rid of the pair of hydrogen bonds that are formed between the side chain and the backbone elements of the NRR. Now, when we stimulate with 100 piconewtons, our upper limit amount of tension application, we can activate expression of the receptor to the same degree. So we essentially max out the reporter here. More interesting is that which falls in between. Okay? So cells that are grown on 12 piconewton TGTs, remember 12 is not enough to activate sNRR. And so the expression of the M-cherry reporter follows almost exactly with that of the no ligand control. But when we challenge the mutant receptor, cells expressing the mutant receptor now with the same amount of tension, 12 piconewton TGT, we're beginning to get some level of receptor activation. So this gave us an indication that mutating the receptor at this position or eliminating this pair of hydrogen bonds mechanically weakens this protein domain, okay? Because now the same amount of force that before gave us nothing is giving us a little bit of expression. So a small fraction of the ICDs have been liberated and have translocated to the nucleus. When we increase the amount of force that's applied by varying the TGTs, the same thing follows. So for 43 and 56 piconewtons, 54 piconewton TGTs, in both cases, the mutant receptor gives us more of a reporter activity. So in this case, what we've done is we've mutated the receptor, and the receptor now can detect a 12 piconewton stimulus, but it's barely able to distinguish between a 43 and 56 piconewton stimulus, 54, or 56 for that matter, stimulus. Okay. Now, phenylalanine is great. We've eliminated this pair of hydrogen bonds that exist here. Next, we wanted to probe the role of this actual aromatic ring here. And so we made another mutation where we changed this specific position right here. We got rid of the ring. And now at this position, we've encoded alanine, so Y186A. And so what we see now when we eliminate both the hydrogen bonds and the ring, now we see a receptor that is really trigger happy, something that 12 piconewtons is able to activate to a very strong extent And now it really can tell no difference between 43 and 54 piconewtons. And we think we have a rationale for that. But since my students spent a lot of time on the following animation, I'll just play it again. Using the same similar strategy in that we were inspecting the protein structure and making educated guesses about how to destabilize the protein, we've generated a family of receptors, all with different mechanical sensitivities. And here we're plotting the reporter expression levels in response to no ligand controls or the various TGTs or mechanical stimuli as shown here. And you can see that different cells respond to different degrees depending on the amount of tension that we challenge them with. We identified one receptor that we think may be mechanically stronger, but resolution of the probes is not such that we can make a really confident argument. Nonetheless, this particular mutation here at this position, we can say that it definitely didn't weaken the receptor. Okay. So next, what we wanted to do was to actually use these for something. So we applied these receptors together in different ways to ask whether we could engineer cells now to sense forces and then do something cool in response. And so going back to that example that I brought up from earlier, where stem cells are able to sense the physical properties of the extracellular matrix and differentiate into bone, neurons, or muscle cells, we wanted to sort of mimic this strategy. And so we want to use now the force of TGTs or the tension that's applied via TGTs in order to direct a differentiation event. Okay. So knowing that cells can sense forces and activate gene expression, change metabolism, et cetera, we want to fine tune this relationship and ask whether sNRR, NRR, and all these different mutants could be used to set those thresholds to different degrees. Okay. So the example that we worked in, however, was the differentiation of fibroblasts into muscle cells. And this can be carried out in a fairly straightforward manner by turning on the expression of a master myogenic regulator called MyoD. And whenever MyoD is expressed in fibroblasts, over time, you'll get a lot of cell-cell fusion, and then you'll get the formation of muscle cells, the expression of myosin heavy chain, so on and so forth. Okay. So what we did now is we took our receptors of varying mechanical stabilities. They have this extracellular domain that detects our synthetic ligand. In this case, all of the synthetic ligands are fluorescein, and we attach the intracellular domain to a tetracycline transactivator. Doesn't matter what it is. All you need to know is that force is applied across this receptor. And then once the ICD is released, we liberate this transcription factor that can now turn on our recombinant version of P65 MyoD. P65 is a very strong transcriptional activator. So when we have signaling, we get this super MyoD turned on in these fibroblasts. Okay. And we compared, again, responses from two cells expressing two forms of the receptor, the regular NRR. So these are fibroblasts in which all the fibroblasts are expressing the NRR containing SynNotch receptor. And these are cells grown in the absence of ligand. And we do two stains. We stain the cells with DAPI, which highlights the nucleus. And we also stain the cells for myosin heavy chain, which is a marker of the myoblast state of the cells. So these cells, these are fibroblasts here. They're not expressed in myosin heavy chain, and cells are there because they have DNA. Now, when we grow the cells on ligands tethered to surfaces fluorescein, attached via 12 piconewtons or 54 piconewtons TGTs, in both cases, we get differentiation, the formation of these muscle-like cells. You can tell because they're expressing the myosin heavy chain marker and they have multiple nuclei, and both TGTs are able to activate that pathway. Now we have a different cell type though. So this is the cell type that I like to say is the older Xth year assistant professor muscle cell type, in that it really doesn't want to become muscle anymore. Okay, so now we have cells that expressed our strengthened SNRR-based receptor on the absence on control surfaces without ligand. They're all still fibroblasts. And in this case, 12 piconewton TGT, that stimulus is not enough to drive the formation of muscle cells. But thank God, if these cells put in a little bit more time at the gym, you hit them with a little bit more force, they get something that kind of resembles muscles, like the who are we kidding? There were never muscles in the old days, but you know what I mean, right? So now if we apply more force to these cells, they're able to differentiate into a myoblast-like state. And so what we've generated now are two cell lines, fibroblast cell lines, that can feel the difference between 12 and 54 piconewtons and then respond accordingly. Okay, that's just a closer up view. But revisiting this sort of schematic, we realized that nature has much more sophisticated than our synthetic designs here. What nature's done is actually was able to take mesenchymal stem cells and have those cells make a decision with three outcomes, depending on the stiffness of the extracellular matrix, the Young's modulus here. So very stiff matrices, you get bone, soft matrices, you get brain, and then medium stiffness, you get a myogenic fate. Okay, so this stem cell here is able to discriminate or respond, becomes a myogenic fate, goes down in myogenic lineage only in response to intermediate values of the ECM stiffness. So we want to mimic this band gating, this Bandpass type behavior. Okay, so essentially we were challenging our system here, asking whether we could use these systems to guide cells to not only sense different amounts of forces, but to make decisions on discrete amounts of tension. So for instance, could we direct cells to turn on MyoD in response to some amount of force that lay in between two windows? And so have the cells stay fibroblasts at a lower level of tension or fibroblasts at a high value tension. So have the cells become, carry out their specific biochemical activity or transcriptional activity only in response to intermediate tension levels. Okay. So in other words, can we program these cells in a Goldilocks-like manner to find just the right amount of tension that makes them ready to carry out whatever function? And so in order to do this, we combined two receptors. We combined one of our weak domains, as I'm calling it now, so the NRR containing receptor, and we used it in combination with one of our strong domains, so the SNRR containing domain. In this case, the receptors contain the same extracellular domain, that fluorescein binding domain, and we varied the intracellular domain. So there are two distinct transcription factors now, a GAL4-based transcription factor linked to the weak receptor and a TTA-based transcription factor linked to the strong receptor. And the significance of this is that when you activate one receptor, you'll get one gene, and then the other receptor turns on a second gene. Okay. In order to get that gating behavior, what we did was we linked the receptors in a way such that a low amount of tension, a first threshold here, would activate one receptor but not the other. And that would lead to the expression of a GAL4 dependent reporter gene, in this case, CFP or M cerulean. And at these intermediate tension values should be enough to activate the weak domain, but not sufficient to activate the strong domain. Okay. And then when we would raise the tension level now, in order to engage that second arm of this circuit, what we did was we linked the activation of this receptor with two things. One, the expression of a red fluorescent protein, simply so we could see it, but also, in addition to that, an inhibitor, something that would turn off the expression of that first arm. And the inhibitor that we chose to use was a microRNA here that is linked with the expression of the red fluorescent protein, and that microRNA goes around and chops up the transcripts, the mRNAs that encode for M cerulean. So when we get this second receptor, second arm activated, we would expect only to see red fluorescence, and the green fluorescence should be diminished. And that's what we saw. So in the absence of ligand, both arms of the receptor of the circuit are off. When we stimulate the cells with 12 piconewtons of tension, we turn on that first arm, which leads to the expression of CFP. And then when we stimulate the second arm of the circuit with a really strong high level of tension, we get RFP or red fluorescence. And that red fluorescence is accompanied by the knockdown of the green fluorescence emission. But in addition to programming things, we also wanted to use the system to learn something about biology, or show that we might be able to learn something about biology at least. And so what we did here, this is actually hot off the press. I haven't rehearsed this yet, so it might be, we'll see how it goes, okay? And that bit right there was not rehearsed. But in any case, we next asked whether we could use this to study something about biology. And so we looked into the future. We asked, what is it about natural notch that requires mechanical force for activation? And it turns out that endocytosis and the actin polymerization that's required to form an endocytic vesicle is the force generating entity that delivers mechanical tension to the receptor to activate the signaling in the adjacent cell. And I say that we looked into the future because this paper actually has not been published yet. I'm just kidding. I ran into this yesterday. So that bit was rehearsed out loud in front of a mirror. I'm just kidding. I don't know. This is very awkward. I'll just keep talking. Okay. But if you look at the first sentence, a few sentences of the introduction in this paper that uses cryo-EM tomography to characterize endocytic pits and look at the actin polymerization that surrounds these vesicles, the second sentence cites this previous work that's defined a range of force that is applied by endosomes, so 1 to 10 piconewtons. Okay. And as I mentioned, endocytosis is what actually delivers mechanical energy to Notch in order to break that domain and liberate the transcription factor sitting at the plasma membrane. And endocytosis is actually required for activation of Notch. So if you mutate the IC such that it's less efficiently endocytosed, you don't get any signaling. If you inhibit endocytosis, you don't get any signaling. And so that endocytic event, transendocytosis, as it's called, pulls on the protein in order to stimulate the release of transcription factor. And the absence of insufficient force, or in the absence of endocytosis, no tension is applied across this bond here, and so no signaling ensues. Okay, so at this stage, we actually wanted to apply the system to measure the force of endocytosis directly between cells. So previously, people had measured these using beads, single molecule techniques, et cetera. But endocytosis and Notch signaling happens at cell-cell interfaces. So we wanted to ask whether we could actually observe, quantify, and measure how much force is being applied by a single endocytic event to stimulate signaling in a neighboring cell. And so we replaced our TGT-based tension delivery system with a cell-based tension delivery system. And in this case, we're mixing two cell types together to mimic the signaling that happens at cell-cell interfaces naturally in Notch. And so we mix our cells that express our receptor with cells that express now this synthetic ligand. Okay? And the synthetic ligand is an anti-biotin antibody fragment, previously published by Brinkman et al. At Pfizer in Europe. And so this antibody fragment now is fused to a SNAP-tag domain so we can see it. And then it contains the intracellular domain, or the endocytosis signal, from a natural Notch ligand called DLL1. Okay? And when we express this receptor in cells and label the SNAP-tag with a cell impermeant SNAP-tag dye, we see that the surface of the cells light up, and we also see these internalized vesicles. Okay? And so the dye that we use cannot cross the plasma membrane, and the reason the formation of these vesicles actually arises from the endocytosis of these the copies of these proteins as the ligand is internalized via this DLL ICD region in the cytoplasm. Okay. And then we mix the two cell types together, our ligand expressing cell, our receptor expressing cell, and then we find a way to engage the two cells so that they can be mechanically coupled. And in this case, we use a simple bifunctional molecule, something that has fluorescein on one end and then biotin on the other end. This molecule can now bridge the senders from the receivers together, and then now link the cells or mechanically couple the two cell types together. Okay? We grow the cells in these various microwells. Sorry, this should say biotin fluorescein here, not docs. So really really hot off the press. But we grow these cells in these microwell chambers. Initially, we remove this microwell chamber. We allow the cells to grow into one another. And what we're looking at here are cells that express the ligand, as indicated by their reactivity with the SNAP-tag dye. And then cells that express the receptor are marked with BFP. And when we add that bridge, we can induce signaling across this cell-cell interface. In the absence of that bridge, the cells are not mechanically coupled, and so we don't get any reporter expression. So we can do it in this nice fancy pattern, micropatterning fashion, or we can simply mix the cells just together randomly. Okay. And so just to confirm that we are inducing signaling and expression of our reporter in a ligand-dependent way, we add biotin fluorescein at these varying concentrations. Remember, this is a bifunctional molecule, and we get actually very sensitive signaling because we think that the binding of one arm localizes the second arm to the cell-cell interface here. And we can express HB M-cherry expression in a way that can be saturated via the hook effect, just like any other bifunctional molecule. Okay, so we applied the system now to measure endocytosis, or the force that is applied by endocytosis directly at cell-cell interfaces. And I'll remind you, we have all these receptors that we fine-tune across this board here, and that the force of endocytosis is about 10 piconewtons. Or let's just keep rounding up and say that endocytosis is about 12 piconewtons, somewhere not so far away from 9, 10, now 12. Okay? So we expected, based on our initial TGT data, that some of these cell receptors should be activatable by endocytosis, and other ones, the mechanically stronger ones, should not. And it's in particular the ones that have low reporter activity in response to 12 piconewton TGT. Okay, so I'll walk you just through a bit of data here, the first of which will compare the natural NRR containing receptor against a SNRR receptor, our weakest SNRR receptor here, the SNRR receptor that contains the mutation of arginine at position 103 to alanine. Okay, so first of all, when we grow the cells with just not the receptor cells with HEC control, so cells that are not the sender cells, and we add biotin fluorescein, we see nothing. Cells that express the NRR containing receptor, when we add biotin fluorescein in the presence of anti-biotin senders, we get activation of M-cherry. And it turns out that our weak SNRR receptor, the R99A, we changed the numbering convention along the way, which is just the worst thing to do. So all the numbers are different. But in any case, this is R103A, the weakest receptor, still activates due to endocytosis. Okay. Next, we wanted to test this Y186A, which was originally called Y49A, but it's that one that I showed you earlier with the structure and the various TGT. So this is the weakened receptor. We eliminated the aromatic ring, the pair of hydrogen bonds, and we have an alanine at position 186. And 12 piconewtons stimulates activity to some extent. So when we look at those cells in the presence of biotin fluorescein against anti-biosenders, we get some amount of signaling. And signaling is notably lower than what we observed with NRR and R103 mutant. Okay? And so just to confirm, however, that this signaling was reduced due to the mechanical, that this receptor represented a mechanically stronger domain than the other two that I'm showing here, we also evaluated a double mutant in which the Y186 mutation was further destabilized by a second mutation on the NRR. And in that case, we get a little bit more signaling, at least so far, it now seems. Really hot off the press. Okay, so we also wanted to compare, for sanity's sake, receptors that we expected not to be activated by endocytosis, okay? And in particular, we looked at Y186F, which, as you can see here, in response to 12 piconewton tether, exhibits about the same amount of signaling activity as SNRR. So not much signaling activity is occurring in response to 12 piconewtons of tension here. And so we grew these cells in the presence of anti-biotin senders, added that chemically inducible mechanical coupler, and at the same concentration. And in this case, we weren't seeing any signaling across these cells. So what we think we see here is that we're able to now apply these receptors as sort of a genetically encoded AFM that allows us to measure the forces across cells. We know from previous work that endosomes can apply about up to 10 piconewtons of force. We calibrate these receptors with single molecule TGTs. And what we see at the TGT level agrees more or less with what we see at the cell-based level. I'll end with thanking the people who did the work. And most of the work was carried out by Chris Sloas, who again has now moved on from the lab. But Remy Peace, these people worked together as part of a Notch team. Remy Peace worked on the anti-fluorescein aspects that we fused to the receptor, and we have some other applications going on there. Jeff McMahon, who worked on the antibiotinamide ligand aspect of the work. Jeff's now nearby at Asimov. Remy's a consultant downtown in Boston at Clarion. And Jeremy Tren and Alex Marzilli, these two folks here, are still in the lab, and they carry out the cell-cell signaling aspects of the work. And they're actually, if you want to come visit them, they're going to be there for the next 8.5 years. So come by and say hi anytime. But of course, we have other efforts going on in the lab, and I didn't have time to talk about them. That's it. I hope I left time for questions.
Questions? Question about the bandpass filtering. So if you wanted to do the mesenchymal thing of going to multiple states, is that just a question of integrating multiple bandpasses in a cell membrane?
Yeah. I think it's possible. I think it'll probably require further engineering. We'd hope that these individual receptors would act like switches. It turns out that their activation is more of a smooth curve. So varying levels of force activate the receptors to different extents. So I think that more defined decision-making processes could be achieved either by evolving a switch-like activity or by exploiting the various receptor thresholds to have the cell now interpret, all right, this one's on to this extent and that one's on to just this extent, and then having that type of logic. But yeah, so I think Alex and Jeremy will be on it for the next 1.5 decades.
John, that's awesome. Thanks. Great talk. I have a lot of questions, but in the last part, did you mention what is the force required to break the fluorescein anti-fluorescein interaction?
Oh, yeah.
I assume that's stronger than all the other interactions.
Yeah, that's a really great question. We chose fluorescein for the way I tell it now, 2 reasons. One is because the original TGT method actually required like purifying the protein for the natural ligand and then coupling it, which is fantastic technique in case people are logged in. But we're a little bit lazier, and we wanted to just order the TGT from the company. So fluorescein can be a built-in oligo that IDT will dye that oligo that IDT will attach to dye, attach to an oligo and send you overnight. Fluorescein, it turns out, is one of and its interaction with the antibody has been mechanically well characterized. Yeah. And so, and not just this antibody that we use, its cousins as well. This antibody takes about 80 piconewtons to break. And I think it's, I have a demonstration. Okay. If you think of this bifunctional inducer, all of these things sort of are linked together, like links in a chain. And whichever bond breaks first will define whether signaling happens or not. And and so we like to think of these things as sort of genetically encoded tensiometers. I don't even know if the camera's on, but you could imagine these components here are measuring the force across these bonds, and that transmission of force will be limited by the strength of the adhesive holding this thing together. So whatever holds our groups together, they got to be stronger than the
that was kind of the source of the question, because I wanted to make sure that it was stronger. And I mean, another interesting aspect of it, right, is that these are all supramolecular interactions, and they presumably have some dynamic component as well. Maybe the antibody fluorescein binding is, has a really low dissociation rate content, so it's essentially like static. But what is this? If there's rapid exchange, then actually what you'll measure will depend not also on the rate of the measurement, right? If you do it very slowly, you may see, but there's actually no bond. And if you do it very quickly. So
this is great. We've known each other for a very long time, and our discussions are always like this. Just kidding. So and this was not planted at all. I'm just kidding. But you mean you don't? You don't? No, I'm just kidding. So the one of the criticisms of AFM is that AFM works in a regime that's beyond the natural loading rate. Natural loading rate in itself, the rate at which cells can apply force, is controversial in itself. But that antibody study actually evaluated the force rupture of the bond across many different loading rates. Now, another aspect is that the K-off actually turns out to be the key parameter that most closely links mechanical rupture, the mechanostability of two interactions, ligand and receptor, against force. And so through something called the Bell-Evans model, I think. But force, of course, we know force is a vector. And so it will depend very much on the direction of pulling. And so the antibody in this case is bound to the ligand, and we have a specific orientation which we can predict that the pulling is going to occur. But if you take just ubiquitin, for instance, and you pull it from one, across N and C terminal versus other ones, you get all different types of unfolding forces. But one theme that we are learning from these studies, I didn't have time to talk about it, is that we think that a feature that makes interactions mechanically strong is some, loosening plug or this mechanical latching effect in the actual three-dimensional conformation of the protein. So proteins that are the most mechanically strong proteins seem to have very high affinity interactions that are low K-off, but also some steric bulk that disfavors the dissociation of those two components like that. And people know that from natural Notch, but also we think we know it from our antibody-based designs too. We think we know a lot of things. We'll see. Yeah.
Well, let's thank John again. That was an excellent talk.
Thank you.